very much for the invitation. And I'd also like to thank you all for running a session in English. I really appreciate it and realize that it must be a hardship for you, and hope that someday I can learn at least a few words of Korean to make it properly. I enjoyed my first visit very, very much, and it's wonderful to be able to visit my former student in particular and see his department and his colleagues. So what I'd like to tell you about is some work that we have done looking at proteins on lipid surfaces. So these are not polymer materials, but proteins, which are biological macromolecules. And I hope their ordering processes will be of interest to you. So I'll spend some time giving you some background on order-disorder transitions, or crystallization phenomena that we are interested in on colloidal and polymeric length scales. And then I'll give you some motivations from nature that we may be interested in proteins in particular. Then I'll tell you about some of the work on lipid monolayers that we've done over the past 5 or 10 years, and tell you how we can manipulate the crystallization using classical colloidal approaches from colloid science to change the interactions with ionic strength or pH or the solution properties. The nice thing about proteins, however, is unlike colloidal particles, here we can do point mutations and change single amino acids and change the interaction between proteins in a very specific way. And so I will talk about how we did some work using mutations. Finally, I'll tell you about some recent work on putting crystals on vessels, liquid-like structure to an ordered structure. And many of you may be familiar with colloidal crystals that have nice rainbow colors because the colloidal particles form an ordered array separated by several colloidal spacings, and they diffract visible light. What's nice about these systems as compared to molecular systems is that they have experimental conditions we can manipulate to cause this ordering transition. So with a colloidal suspension, for instance, we can change the volume fraction of the particles, the ionic strength. If we have polymeric micelles, we can change the molecular weight of the polymers. In the protein case, we can change the pH. This, of course, is much easier than looking at, say, an ordering transition in metals where you have to go to high temperatures. At a lot of length scales, and this is actually was on a calendar when I was a graduate student, and I kept it for some reason. I was interested in ordering way back when I was a graduate student. It's from the Sierra Club calendar and showing penguins on their breeding grounds. But any system where you're trying to maximize the space between your neighbors, you find you naturally form an ordered array. This happens amongst children doing their exercises on a playground or amongst these penguins. You can make a lot of remarks about the level of order and then the dislocations and then the order parameter causing all the penguins to face in one direction because of the sun on their backs. But then there's always an odd character in every group. So I won't go on with the social commentary. Ordering is an entropic process, and you can make some analogies with entropy in social groups. If we go to slightly smaller length scales, there's some beautiful work from a few years ago by George Whiteside and co-workers, where now they have millimeter-size objects. These are polydimethylsiloxane objects that have been sliced from a cylinder of a certain shape. So really, you're just looking at the geometric effects. These are floated at an oil-water interface, and their sides have been manipulated to either be hydrophilic or hydrophobic. So all you have here is the gentle forces of capillary action with these objects, I'm going to blind myself, floating at the oil-water interface that causes these to self-assemble into ordered arrays. And so this is a very nice example of how very gentle forces and shape can affect organization. Here, all 6 sides of the hexagon are attractive, and here only 3. And so with the same shape, you get 2 different structures. Here you have some without attraction that get expelled from the crystal lattice. We move down to yet a smaller length scale and look at protein ordering in 2 dimensions. But again, they're fairly weak forces that are causing this organization. There's several reasons you may want to do this, and the biochemists really want to organize proteins to determine structure. And so Roger Kornberg really developed this 2-dimensional organization, along with Helmut Ringsdorf, some years ago to try to crystallize proteins that weren't readily crystallizable in 3 dimensions. After much work, Kornberg's group was able to get this pole 2 structure you may have seen in Science magazine a couple of years ago. This was a huge amount of work in 2-dimensional ordering, but it gave them a very useful and important protein structure. You may want to organize proteins for templates or biosensors or other arrays. And for us, I'm also very interested in understanding how a protein coat influences a natural system such as a cell. So an example of the cell membrane is an S-layer protein on archaebacteria. This is a picture of an archaebacteria cell surface, which has an organized array of proteins that are attached to the membrane by electrostatic binding. It's thought that this ordered array has a role in filtering or sieving materials that go through the membrane because it has a very well-defined pore size. It's also interesting to ask how much mechanical integrity this protein coating gives to the cell, in addition to the other structural components in the cell. So we take a simpler view, a simpler system, and put an organized array of proteins on the outside of a plain lipid vesicle and look at the difference in the mechanical properties between a plain lipid vesicle, a lipid vesicle coated with disorganized protein, and one coated with protein crystals. And that's what I hope to show you. Another very interesting application or manifestation of ordered proteins comes in these bacteriophage that attach to a cell surface and then inject their DNA into the cell. This injection apparatus is a cylinder that has an ordered array of proteins on it. Upon docking with the cell surface, something happens that causes the proteins to go from one crystal structure to another. In our system, we see we can get crystallization transitions from one structure to another by a change in pH. You might imagine that certain ions or calcium ions or other physical properties can be used to invoke a crystalline organizational change. And so when you see a large effect of the function, obviously this crystal change causes a collapse of the cylinder, which injects the DNA through the membrane. It has a very important biological function, but it may have a very simple physical background. So we take the same approach that I showed with the colloidal system and look at how to manipulate protein-protein interactions through pH, ionic strength, knowing their electrostatic interactions, in the presence of sugars, or making mutants of individual amino acids on the proteins. We ask how this affects their molecular organization or crystal structure, and how it affects their crystal morphology, stability, and now the mechanical properties on the outside of a vesicle. The model system we've been working with is streptavidin. Many of you may be familiar with streptavidin. It's a tetrameric protein. It has 4 subunits labeled in different colors here. Each of the 4 subunits can bind essentially irreversibly to the vitamin biotin. So streptavidin has become very popular as an adhesive, essentially, as a means of attaching one thing to another. You'll find it for attaching DNA to a colloidal particle, for attaching a lot of biological macromolecules to surfaces, or in various separation applications. You can take advantage of the strong binding for biotin. What's important for us is that there are 4 binding sites for biotin in this tetrameric protein. Biotin is shown here in green in 2 of the 4 subdomains. And in this protein, the tetramer is like this so that there would be 2 binding sites on the top and 2 binding sites on the bottom. So when we attach streptavidin to a lipid, we use a biotenolated lipid. That will attach streptavidin to a lipid layer, and it leaves 2 open sites. This has been used by a number of people to take a layer of streptavidin as a building block for a multi-layer structure. We won't be doing that, but I'll show you that there is an effect of attaching biotin to the streptavidin. So what we do is quite simple to draw, and of course difficult to do because surface science is always fairly challenging. But we take a monolayer of lipid, put in 1/10 biotenolated lipid, put the protein into the subphase, compress to the desired surface pressure, put the protein in the subphase, it attaches to the lipid membrane, and then forms crystals. So if you look at a drawing to scale, you would see streptavidin here with a number of lipids above it, about 40 lipids per streptavidin molecule by area. And 2 of them will have biotin and be attached to the streptavidin. So this organization serves 2 purposes. It brings the streptavidin to a higher concentration in the monolayer, and it orients it, so it reduces the number of degrees of freedom. And streptavidin was actually first crystallized in 2 dimensions when it wasn't crystalline in 3 dimensions because of this reduced degree of freedom. It orders quite readily and forms crystals quite readily. I'll note that we also put in kind of a filler protein. These gray molecules are avenine. Avenine is a molecular analog of streptavidin, but it has a sugar coating on the surface. Sugar, it's glycosylate, it has sugar groups on the surface, so it does not crystallize. So you can think of the streptavidin as the crystal, the avenine as the liquid or the solvent in 2 dimensions. We then look at these crystals with fluorescent microscopy to see the structure, the morphologies after showing off in these little carbon pictures. And then we take and transfer these crystals to electron microscope grids and look at their crystal structure. We focused on changing the subphase to manipulate the interactions to change the crystallization behavior, and then we did mutagenesis to be more specific with our interactions. So Sue Wong, a student of mine several years ago, found fairly quickly that if you change this pH of the subphase, you got a wide variety of crystalline morphologies reflecting different crystal structures. A previous student, Amber Koo, had already noted that we get very anisotropic crystals. In particular, around neutral pH where we started, we always get rectangular or large X-shaped crystals. And the crystal structure at this pH we knew was square symmetric. It's a square protein and made square crystals, yet they grew faster in one direction than the other. This is due to the fact that the 2 biotin sites are bound with biotin and 2 are empty. That very small conformational change upon binding actually changes the protein-protein interaction enough that you get a faster growth in the biotin-bound direction than in the unbound direction. I didn't want to go through all that here, just to tell you that shows you how weak the forces are and how delicate they are, that the binding actually gives you a change in the energy for the crystallization and gives you anisotropic crystals. You also get these dendritic growth patterns because we have transport-limited crystallization. The streptavidin has to diffuse through the avenine, and you get a concentration gradient set up, much like you have a temperature gradient in a snowflake. In any case, we found we could get these different morphologies, and indeed, if you do the electron microscopy, electron diffraction, and then determine density maps for the proteins. And these are done with a stain, so they're not very high resolution, but they're good enough to determine the structure that we need. We find that we have different crystal forms at different pH. So it was well known that this square crystal existed at high pH and neutral pH and higher. It was known also, just before we started working on this, that at low pH you got a different crystal structure that was a higher density and results in a different morphology. What Sue Wong found was that there's a new crystal form around the isoelectric point, around pH 5.5, that now has a structure that has P2 symmetry, 2 proteins in the unit cell, and has this kind of interesting shape. It's kind of like one of these with every other row translated by 25 angstroms. What's interesting about this shape is that there are 2 ways you could draw it. It's kind of like a Tetris object, one of those, if you played Tetris, that computer game. And you could draw it in either direction. Again, the biotin binding, we believe, breaks the symmetry and favors this inverse S rather than the S shape, and gives you the opportunity now to have this kind of packing arrangement. So the different crystal structures that we have then are reflected in different morphologies. These square crystals give us rectangular or snowflake-like crystals, morphologies. These P2 crystals also give snowflake-like morphologies. And these higher density P1 crystals give these needle-like morphologies, very densely packed. Whenever we have these inverse S-shaped crystals appearing, we always have a mixture of this Tetris shape, this inverse S-shaped crystal, along with the P1 crystal. It doesn't take too many of these packing in these rows in between layers of P1 to give us this kind of a morphology. So if you look in detail at the molecular packing in these kinds of inverse S-shaped crystals, you see this kind of striped packing, and a little bit of P2 serves to give you that curvature in the morphology. This will be important later when we do our mutant studies. So we were concerned that when we changed the pH, we're changing a lot about the interactions. It's just kind of treating the proteins like a colloidal particle, and changing a bunch of amino acids because we're at a different pH. Their interactions change, and sure, you get a different packing. We wanted to try to understand that a little more clearly, so we did some modeling to look at how the proteins were packed in detail relative to their preferred growth direction, their rapid growth direction, and then to look at what amino acids were actually interacting in this case. We focused our attention on the P1 crystal because it's the most densely packed and gave us the most interactions. And if you look at a model of how they're packed, this being the needle-like direction, again you see biotin bound and biotin free. In this case, the fastest growth direction is kind of along the biotin free subunit direction. We looked in more detail again and tried to identify which amino acids were changing their behavior around pH, between pH 7 and pH 4. We found that aspartate in particular was a good candidate. Both the N groups are also important, but they were not good candidates because they're not well resolved in their molecular structure. So we decided that if we could change this aspartate 36, we could manipulate the interaction in this closely packed structure and see if we understood what was going on. So you'll see there are 4, end on end, there are 4 interactions. If you look on the side view, remembering that it's a tetramer, we have this kind of an arrangement. At pH 4, these aspartate groups should be attracting one another through some hydrogen bonding, and at pH 7 they should be repulsive through electrostatic repulsion. So we decided we would try changing that amino acid to a few other candidate amino acids. So our wild type would be the aspartate. Asparaginine would provide hydrogen bonding, strong hydrogen bonding in that direction and give us an attraction. Alanine would give us basically no interaction with a very small amino acid. And we could put lysine on to give us a steric repulsion, positively charged and long side group. So by changing from our wild type to these others, we could try to manipulate the proteins that form. And indeed, it works. I like to just focus the attention on pH 4. Sue Wong did a tremendous amount of work on all these mutants, and this is a very fast overview. The wild type forms P1 crystals in these needles. Interestingly, when you add the hydrogen bonding, you form P1 crystals, but in a different morphology, this kind of asymmetric X or angel shape. When you take away the interactions, you form very nice P1 crystals with very fat needles. They grow much more regularly. And when you put on the repulsion, you cannot form P1 crystals at all. You prevent that interaction and form the lower density crystal structure. So this was very gratifying. This is what we expected. We were interested in this new morphology, and you can understand how it exists by thinking about what you've done in the interactions. The preferred growth direction was along this direction. We added an extra attraction along the head to tail between the proteins by adding this asparagine to give us a strong hydrogen bonding. That increased interaction in that direction gives you two rapid growth directions, so now you get this kind of shape that's no longer needle shape, but is growing off in two directions, and matches very well the geometry of the packing. Interestingly, if you spend, if I had more time, if you let these sit for 48 hours, they actually anneal into these kinds of structures. So these are grown rapidly in two directions, but not as stable, and they form more perfect crystals over time that look like needles. What I'd like to do here then is to remind you that we had these types of S-shaped morphologies when we had a mixture of P1 and P2 crystals. So we decided to do a kind of outlandish experiment. What if we mixed wild type with this mutant? And it wasn't clear at all that if you take two proteins, one that's mutant and one that's wild type, whether they would co-crystallize, whether they would separate, whether they wouldn't crystallize at all, or whether they would crystallize into P2 domains and P1 domains and give us this kind of structure. And sure enough, student Sammy Farah did this, took wild type, mutant, mixed them together, and this is the only time I'll do this. You get the inverse S-shaped morphologies that are coexistence at P1 and P2 lattice. So now you can do a molecular engineering and make a mutant and form a different type of crystal from a mutant, plus you can mix it back with a wild type and form a structure that's like the wild type forms at pH 5.5, but here we're at pH 4 with totally artificially manipulating this. He did all the microscopy to show that indeed we had P1 and P2 co-crystallizing in these, and it was just something that we were kind of excited about because it's a new way of looking at these types of systems. Well, to work with lipid vesicles, we can't work at the usual conditions. Our usual conditions for these are at 500 millimolar sodium chloride, which has a very small Debye length, very strong electrostatic screening. Vesicles are not stable under those conditions, and you have to go to very low salt conditions to form crystals. What we found, at first we were concerned that we wouldn't form any crystals because of the long electrostatic repulsion. In water that is the case. For some reason in the presence of sucrose, we do form crystals, except at the neutral pH, pH 7, we can't form crystals anymore. So when we're working with vesicles then, we will expect to see these types of crystals. If you add salt back to the system, indeed you go back to the crystals that you would form before. So interestingly, at low salt, these are all C222 crystals, and they don't form at pH 7. They only form nearer to the isoelectric point of the protein where electrostatic interactions are smaller. But we wanted to grow these onto vesicles to look at what they did to a vesicle structure. We form these so-called giant unilamellar vesicles by an electroformation process with dried lipid on electrodes. And we can vary the membrane composition just as we do in the monolayer experiments. And we add a biotinylate lipid to the normal lipid. And then they form these nice kind of 50 to 100 micron size vesicles that we can work with. They're suspended in sucrose. They're made in one of them, glucose, and suspended in sucrose, or vice versa, so that they're dense and they settle to the bottom and can be easily manipulated. And also it gives you an index of refraction difference so you can see them quite well. You can see from confocal microscopy that indeed we do get crystals on the outside of our vesicles. These look more or less just like our monolayer systems. Again, at pH 7 we form no crystals, and then at different pH, these are mixtures of streptavidin and avidin again. Here the avidin is labeled, the streptavidin is dark. And you see all the different morphologies that you can find. Still some of these X shapes, still some needle-like crystals. Interestingly, here the needle-like crystals are at the higher pH, and the larger crystals are at lower pH. If you do the electron microscopy to prove to yourself what is on the surface of the vesicle, you see that here you get multi-layers, so you have to sort out, you have at least 2 or sometimes 3 layers when you collapse a vesicle in a EM, but you see that we have C222 crystals, those square crystals, under all these conditions. We were interested in what happened and whether the crystals could have an impact on vesicle shape. So if we have mixtures of streptavidin and avidin, again, avidin is amorphous and never crystalline, streptavidin is crystalline, you see the morphologies. If you have pure streptavidin, we can't do the fluorescence, we don't have any contrast, but in regular microscopy you can see that you can get these football-shaped crystals or these very lumpy, kind of rough and faceted crystals. We believe that at the lower pH we're forming smaller crystal domains that run into one another and give you this kind of roughened shape. At the higher pH you get these needle-like crystals that tend to form along one direction and give you a preferred bending direction and give you this kind of egg shape or watermelon-like structure. So we wanted to be a little more quantitative and determine what would happen to the mechanical properties. And the Zachman group in Munich is expert. Rudy Merkel's group is expert in micropipette work. And so we decided to study these with micropipettes to understand what was going on the surface of the membrane with the streptavidin on it. Here what one does is pulls the vesicle into a micropipette, and as a function of the pressure looks at the length of the protrusion into the pipette. This pressure against length can be translated into a tension and an area change so that you get a relative modulus for aerial compression on these systems. And I'll just summarize all the observations here. The moduli themselves are not that interesting, but the rheology of the membranes is interesting. So more or less the modulus seems to be dominated by the lipids itself. The bare vesicles, avidin-coated and streptavidin-coated, have similar moduli. If anything, they look smaller, but they're probably not appropriately treated as regular viscous surfaces, as you'll see. Bare vesicles are very spherical and smooth. You can see the streptavidin-coated are distorted in shape. Avidin-coated are very spherical and smooth. You can really get a feel for how they behave by looking at their rupture characteristics. So if you watch a regular bare vesicle break, you see it basically just vanishes. So they'll pull on it and then it just kind of goes. Similarly, if you watch an avidin-coated vesicle, again, it's very smooth and spherical, but now you start to see a deflation, and you can actually see the effect of the fact that there's an amorphous protein coat on the surface. So as you watch this thing break, you'll see it crumple more like a plastic bag. Finally, when you look at the streptavidin coating, again, imagine that now you have regions of ordered protein all over the surface, and that will provide some rigidity, and you can see that when you watch this one rupture. It leaves a more rigid surface, and so clearly the difference between disordered protein and ordered protein is there. Then why I think these are very interesting rheologically and difficult to model very quickly is that we see plastic deformation. And so sometimes if you had a streptavidin-coated vesicle and you deform it, it will stay deformed. And so you can see this in this experiment where a small vesicle was pulled in to the pipette and then pushed out, and this thing will be stable for hours then with this little protrusion on it. So now you can think about making all kinds of cute shapes. So I'd like to just summarize and say that taking proteins and attaching them to a lipid layer promotes ordering because it concentrates them, reduces their translational and orientational degrees of freedom. In this case, the ligand binding breaks the symmetry of the molecule, and so we get anisotropic shapes. Now we get dendritic structures from transport limitations that you can model like other crystal growth problems. We can use colloid science type approaches to change the physical chemistry of the solution to alter the interactions between proteins, and the pH in particular, ionic strength, and now adding sugars, changes the interactions and gives you different crystal behavior. If you have solid-solid coexistence, you get chiral structures, and we can be a little more specific in altering our interactions by changing a single amino acid to either prohibit or change the nature of the crystal structure form. And then we can blend our mutants back with wild type and get some interesting shapes back. Finally, if we put these on the surface of vesicles, we see they do impart a mechanical behavior. They change the rupturing characteristics, and they can have a plastic response. And that might be interesting both in the cell biology situation as well as in some few practical applications. I'd like to thank my colleague Cheney Robertson, who got me into this, working with 2D protein crystallizations. Suwong and Sami Farah did most of the monolayer work that you saw, and Paul Ratanavanankunde did the work on the vesicles. I took Po to Munich for a summer where he did all the pipette work with Rudy Merkel and Michael Gropper in the Zakwin group. And actually, I didn't talk about some work on nanotubules we did with Liz Kubalek and Scripps, and I appreciate the funding of the National Science Foundation and the Humboldt Foundation. Thanks very much.